is Richard Jovelin. Richard is joining us from France via the US into Canada. So he did his undergrad in Lille, France, master's in Paris in evolutionary biology, and then moved into the US to Eugene, Oregon, small place, he said, to do his PhD in evolutionary genetics. And did his first postdoc at the University of Toronto here under Asher Cutter, and studying genome evolution, molecular population genetics in nematodes, of which he's going to speak to us today. And he now works as a postdoctoral fellow in the Ontario Institute for Cancer Research under Philip Awadala. So Richard, just switch you out. There it is.
Right, well, thanks for coming. So, so this is a short talk, so I'll try to fit in 10 minutes. So describing, I will succeed in 10 minutes. No choice. So describing patterns of genetic variation is essential for multiple reasons to understand how genetic variants contribute to disease and to understand what makes species unique. But to fully understand these patterns, we must take into account past geography, population history, eventually lifestyle of the organisms in question, and their ecology. So I want to start the talk by showing you this graph to present you with the null problem in evolutionary genetics. And what this graph is showing here on the x-axis is nucleotide variation for a number of species that are ranked here from low to high. Okay. So the question is, why do we see so much variation in nucleotide diversity among organisms? Okay. As a reference, humans are here on the low range, and you can see a lot of variation. You might recognize some model organisms with different species of Drosophila, and until recently, my favorite species, or system, Cinorhabdalis. And I also want to point out that there's also a lot of variation, even within a single genus. So you're familiar with the model organism C. elegans, that with a very, very low diversity. And one is of its relative, C. bruneri, is actually the species or the metazoan with the highest level of polymorphism recorded to date. It's not the champion of polymorphism anymore. There's a paper that was published 2 months ago showing the mushroom species has over 20% nucleotide diversity at single sites. So it's a huge amount of variation. So in this very short talk, I want to present you some of the work we've been doing to try to understand why we observe such a variation in nucleotide diversity within Cinorhabdalis, and what strategy we are taking to combine functional information that is derived from genetics and functional genomics in C. elegans, both with looking at variation within populations for relative species and comparative genomics among species, to ask questions about the evolution of biological systems and gene function. So, as I was mentioning, C. elegans is a famous model organism. It is used in hundreds of labs around the world. And the reason why Sidney Brenner chose C. elegans in the first place is because he wanted a simple organism to study the development of the nervous system. The other reason why he chose C. elegans and not another species is because of a little trick. And that trick is that C. elegans is a self-linked species. So most of the individuals in C. elegans are hermaphrodites that have both function, male and female, and they are obligatory self-linked. They cannot mate with another hermaphrodite. But they do occasionally mate with males that are found in the population very low frequency. And so Cinorhabdalis is a diverse genus. We have a number of species. Most of the species are what we typically think of an animal. They are outcrossing with male and females, and they are shown in blue on the phylogeny. And you can also see that selfing has evolved independently 3 times around the history of the genus. So all the species here are selfing species. Okay. They are also made of hermaphrodites that are obligatory selfing. So contrary to popular belief, and what you might still see sometimes in textbook or papers, C. elegans and Cinorales in general is not really a soil nematode. And in fact, we don't really see it in soil. And when we go out collecting worms, what we collect is rotting vegetable matter, so rotting flowers, rotting fruits, mushrooms sometimes. So this is a picture of the lab that's set up in the tropical forest in South America, where we were collecting a couple of years ago. But we don't need to go that far to collect the worms. And in fact, I collected some of the Cinorales species north of Toronto, and that was the basis of the population genomic studies that I will talk about later. So we done some work to try to understand why we observe so much variation in nucleotide diversity in Cinorales. And it turns out that this mating system differences, selfing and outcrossing, has a very strong influence, and so far is the strongest determinant of nucleotide variation in the genus. So we collected polymorphism information from multiple markers in multiple species, and we constantly see a lower amount of variation for the selfing species, here in yellow, relative to the outcrossing species in blue. And I'll show you the phylogeny again to show you that this is not due to relativeness among species, but is really due to the mating system. Okay. So that lifestyle gives a clear record in the genome. And the first record we can see is a strong linkage disequilibrium in the selfing species. So what these graphs are showing are linkage disequilibrium on the y-axis and the distance between sites on the x-axis. So in the selfing species, C. elegans and TorBUG, for which we looked at variation across the genome, there are very large haplotype blocks on the order of kilobases or even megabases of being chromosomes. So if we compare that with an outcrossing species, again with males and females, the linkage disequilibrium decays very rapidly on the order of hundreds of base pairs. The other pattern we observe in the selfing species is weakened natural selection against deleterious mutations in regions of low recombination. So we surveyed nucleotide variation in the entire genome of one of the selfing, TorBUG, and this is showing you the first chromosome, chromosome 1. Chromosomes can be divided in different domains based on their recombination rate. So recombination is low in the center and much higher in the arms or the tip of the chromosome. So generally, we observe lower amount of nucleotide variation in regions of low recombination, but we do observe an excess, and this is what the line is showing here. We observe an excess of polymorphism at replacement sites in protein-coding genes relative to synonymous polymorphisms, indicating that selection is weaker to remove deleterious or slightly deleterious alleles in these regions of low recombination. So we can see now why C. elegans may be a very good model for some studies in genetics and understanding development and so on, but why it's not such a good model for evolutionary biologists. Because of the low amount of variation, and because this variation is frozen into larger haplotype groups or blocks, we have very little power to detect instances of positive selection. That is, we cannot really tell the position of beneficial allele that may be under selection. So the strategy we've been using is to, again, look at variation in related species that are, of course, outcrossing, but with some functional information derived from C. elegans. One of the systems we looked at is the evolution of aging. So what is aging? So aging is the deterioration of physiology and metabolism over time. So this is famously in single million a number of years ago, I'm not sure when, but this is what it looks like now. Okay? We're more familiar with this picture again. And of course, organisms age at different rates, and, but sadly, aging is pretty much ubiquitous, and you cannot really escape it. Okay? So that brings a question though. Why did senescence evolve? Well, 2 minutes? Wow, okay, I will rush through it. So why is selection not favoring organisms that can reproduce and live forever? Well, so the explanation, or the evolutionary explanation for this, is that there's a trade-off between maintenance and repair of the stomach and investment of resources in reproduction. So there are different hypotheses for the evolution of aging. I mentioned 2 of them. that are both based on the fact that selection, natural selection, is weaker as organisms get older. The orthogonetic phylotropy model states that a mutation if a mutation is beneficial early in life, it will be selected even if the same mutation is deleterious and may cause a disease, for instance, late in life. The other model is the mutation accumulation model, which states that mutation with late onset, they may be neutral early in life, but may be strongly deleterious late in life, can accumulate in the population because selection is weaker and cannot really remove them from the population as organisms get older. So, I'm trying to give you the information. So why do we care? And what was the relationship between the worms? Well, it turns out that a lot of mutations that increase the lifespan are in this insulin pathway that is conserved among worms and even mammals and flies. Okay. In particular, some of these mutations in these two genes, H1 and DAF2, not only increase lifespan and double lifespan, but they do that at a cost. And the cost is a reduction in fecundity. So these mutations satisfy the antagonistic phylotropy model. Okay. So the normal activity of this gene, in fact, may reduce lifespan, but perhaps leads them will increase the fitness. So if that's correct, we may be able to detect in the genome, in the DNA, a record of these processes. So this is what we set out to do, and we sequence all the genes in these pathways. What we observe is very little variation at this gene H1, almost none. And that's very different from all the genes in the pathway, all the genes we looked at so far. And we're talking about silent site mutations, so mutation that is supposedly neutral, it should accumulate anyway. There are two explanations for this. One is that there is strong purifying selection on this gene. Any mutations occurring on these genes may be deleterious and be removed by selection. The other is actually a different pattern. The pattern of a selective sweep, where this gene may be actually selected for, or some alleles may be selected for. And what happens is that as the beneficial allele is selected, any variation that is linked to this allele will tend to hitchhike with that allele, leading to a reduction in variation around the beneficial allele. So we couldn't do this type of test in the selfing species. We did that in the outcrossing species. What we observed, is decrease in variation as we get closer to H1, and again variation increases as we get further away, consistent with the pattern of selective sweep. Well, I'm going to skip these things. Do I have time to talk about Philip's law? No. This is it.
Talk to you after.
All right.
Yeah,
so
of course.
That's too bad. So we are in this new group, and sorry. So we are. I'm recently moved to the lab of Philippe Larodella, and here's the members in Toronto, the few members in Montreal. We are generally interested in evolutionary medicine, and so this is the type of things where we look at. We want to integrate data on phenotypic variants that relates to human health with some measures of genetic variation. Okay, so I think maybe I'll stop this. This is 10 minutes. As to that. So clearly, it's not a demonstration of what should be done as a 10-minute talk. So keep that in mind.